All right, today we're going to unpack one of the most fundamental tools in all of modern biology, gene cloning. Now, I know what you're probably picturing, sci-fi stuff, right? Armies of clones, maybe bringing back a dinosaur or two. But no, that's not what we're talking about. This is about a real-world technique that has totally transformed medicine, farming, you name it. So let's dive in.
So when you hear that word cloning, what's the first thing that pops into your head? For a lot of us, it's probably a very famous sheep named Dolly. But in the day-to-day world of science, that's just one piece of a much, much bigger puzzle. The kind of cloning that happens in labs all over the globe, every single day, is something else entirely. And you could argue it's way more powerful.
Okay, so this slide breaks it down perfectly. On one side, you've got reproductive cloning. That's the Dolly the sheep method, where you create a whole new organism that's a genetic twin. But on the other side, you have what we call molecular or gene cloning. And this is totally different. This is about grabbing one single gene, just a tiny snippet of DNA, and making millions, even billions of copies of it. And that is the powerhouse we're focusing on.
So you might be asking, okay, but why? Why on earth would we want millions of copies of just one gene? Well, the best way to think about it is like manufacturing. Imagine you have an amazing blueprint, but you need to turn that blueprint into a product you can actually use.
The core idea is actually kind of simple and really, really clever. We take our gene of interest, that's our blueprint, and we pop it into a host that's an expert at multiplying. The classic example is a bacterium. That little bacterium basically becomes a tiny living factory dedicated to our blueprint. And here's where the real magic kicks in. That one bacterium with our gene starts to divide. It becomes 2, then 4, then 8, and pretty soon you have millions of them. And every single time they divide, they copy our gene right along with their own DNA. So now we can either harvest all those copies of the gene itself, or we can let the bacterial factories do what they do best, read the gene's instructions and churn out the protein it makes. The perfect example? Insulin. This process completely revolutionized how we get insulin for people with diabetes.
Now, to pull all of this off, scientists can't just wing it. They need a very specific set of tools. It's like a genetic engineering toolkit. So let's open up the box and see what the 3 essential tools are.
First up, you need a vector. The easiest way to think of this is as your delivery truck. It's typically a small circular piece of DNA called a plasmid, which bacteria have naturally. Its whole job is to carry our gene, our cargo, safely into the host cell, into our factory.
Next, you got to have a way to open up that delivery truck and cut out your cargo, right? For that, we use restriction enzymes. They're famously called molecular scissors, because that's literally what they do. They scan along the DNA and make a clean cut. But here's the key. They only cut at a very specific, predictable DNA sequence. That precision is absolutely critical.
And finally, if you have scissors, you're going to need some glue. That's DNA ligase. Once we've cut open our vector and prepared our gene, DNA ligase is the enzyme that comes in and stitches them together. It forms a permanent bond, sealing our blueprint right into the delivery truck.
Okay, so our toolkit is ready. We've got the vector, the scissors, and the glue. So how do we put it all together? Let's walk through the actual cut, paste, and deliver process.
The whole thing is just beautifully logical. So first, and this is the trick, we use the exact same molecular scissors to cut both our target gene and our plasmid vector. This creates matching ends on both pieces of DNA, often called sticky ends, because they naturally want to pair up. So the gene finds its way into the open vector, and then we add our molecular glue, DNA ligase, to make the connection permanent. Bam! One new, solid piece of DNA.
And this brand new molecule we just created has a special name, recombinant DNA. Makes sense, right? We've recombined DNA from 2 totally different sources into one functional unit. This is our finished, engineered blueprint, ready to be sent to the factory floor.
The last step of this phase is the delivery itself. We take our new recombinant DNA and introduce it to a whole bunch of our host bacteria. The process of getting those bacteria to actually slurp up the foreign DNA is called transformation. Now our little factories are, we hope, armed with their new instructions.
But here's the catch. This transformation process, it's not very efficient at all. Only a teeny tiny fraction of the bacteria will actually take in our plasmid. So the big question is, how in the world do you find the successful ones floating in a sea of millions of cells? This is where a truly clever trick comes into play.
The answer is a method called blue-white screening, and honestly, it's just genius. Here's how it works. The plasmid we use has another gene in it, called Ligase, which, when it's working, makes the bacterial colony turn a bright blue color. And here's the brilliant part. We design our cut site to be right in the middle of that blue-making gene. So think about it. If our gene fails to insert, the blue gene is untouched and the colony turns blue. We don't want those. But if we successfully insert our new gene, it gets plopped right into the middle of the Ligase gene and breaks it. The result? The successful colonies can't make the blue color. They stay white. So all we have to do is pick the white colonies. It's an amazing visual indicator.
So we did it. We isolated, inserted, identified, and now we can grow huge vats of just these white bacteria to make whatever product we need. So now let's circle all the way back to the beginning and talk about the incredible payoff of all this work.
The applications are just staggering. I mean, they're everywhere. In medicine, you've got lifesavers like insulin, growth hormones, essential vaccines, the list goes on. In agriculture, it's given us amazing things like insect-resistant cotton. It's used in food science, in forensics. Yep, DNA fingerprinting relies on this. And it's the absolute foundational tool for the future of gene therapy. This basic cut-and-paste technique is truly a cornerstone of our modern world.
So what started as a clever technique in a lab has become a world-changing technology. Gene cloning has given us an ability to understand and engineer the building blocks of life in a way that was once pure science fiction. Which, of course, brings us to a very, very big question.
So where do we go from here? We've built this incredible genetic toolkit. We can copy, we can paste, we can edit, and we can mass-produce the code of life. For centuries, humanity could only read the book of life. But now, we have a pen. We can make edits, we can add new sentences. So the question for all of us now is, what should we write in the next chapter?